Hello, again, I'm Yun Shu from Central University Medical College. This time, we will discuss human papilloma viruses, HPV. Let's start. These are the learning objectives for you. 1. Describe the general properties of HPV. 2. Explain the pathogenesis of HPV. 3. List the appropriate laboratory tests for diagnosis and know how to treat, prevent, and control HPV infection. First, we will show you a case. A 40-year-old woman goes for a routine gynecological exam. She is sexually active and has delivered 3 healthy children. She has had 6 sexual partners in her lifetime. She is also a pack-a-day smoker. The report of the Pap smear indicates a precancerous change. PCR analysis indicates that cells in the lesion are infected with human papilloma virus. Now, here come the questions. 1. What's HPV? 2. How is HPV transmitted? 3. How can infection be prevented? The first question, what's HPV? The human papilloma viruses are small, non-enveloped viruses with double-stranded circular DNA genomes. At least 100 types has been identified by DNA sequence homology, some of which are potentially carcinogenic genotypes. HPV can be distinguished further as cutaneous or mucosal based on target tissue. As the picture shows, the virus is coated with isohedral capsid. The capsid consists of 2 structural proteins that form 72 capsomeres. Depending on the host cell, these viruses are capable of causing lytic, chronic, latent, abortive, and/or transforming infections. Let's get to know the viral genome more in detail. The HPV genome is circular double-stranded DNA and has a 7.9 kilo base pairs. The HPV DNA contains 7 or 8 early genes, 2 late or structural genes, and an upstream regulatory region. As the table shows, the genes encode different proteins involved in distinct functions. E1 and E2 proteins bind the DNA and promote replication and transcription. E4 disrupts cytokeratins to promote viral release. E5 oncoprotein activates the CGF receptor to promote growth. E6, E7 bind to growth suppressor protein P53 and P105RB and cause immortalization and are associated with human cervical cancer. L1 and L2 are late structure proteins. How does HPV replicate in the target cell? HPV replication is linked to the life cycle of keratinocyte and epithelial cell. 1. HPV infects the basal layer through a very tiny break in the skin. Then, L1 protein attaches to the receptors and initiates the replication. 2. HPV genomes replicate along the cellular DNA in undifferentiated basal cells. 3. Upon differentiation, late gene is expressed and amplified. 4. E6, 7, E4, and E5 contribute to productive replication. 5. L1 and L2 promote virion assembly, resulting in viral release from the upmost layer of the epithelium. What are the consequences of HPV infection and replication? Infection by papilloma viruses induce epithelial proliferation. The HPV types are tissue-specific. causing different disease presentations. The cutaneous squamous epithelial infection can lead to warts, while a mucous membranes infection resulting in genital, oral, and conjunctival papillomas. As I mentioned, different HPV types has affinities for various tissue and hence different manifestations. The table here summarizes the HPV types and diseases they cause. As you can see, HPV can cause skin lesions and mucosal diseases such as benign head and neck tumors, condylomata, acuminata, or cervical intraepithelial neoplasia. A wart is a proliferation of skin cells that regresses with time. The lesion is usually found on keratinized surface of the hands and feet. Condylomata, acuminata are found on external genitalian and perianal areas and are common in promiscuous individuals. Most cases are caused by HPV type 6 and HPV type 11. The genital tract HPV infection is a very common sexually transmitted disease. The infected person is usually asymptomatic. Carcinogenic HPV types infection of the female genital tract can initiate the development of intraepithelial cervical neoplasia and cancer. These pictures show you the different HPV diseases. Picture 1 shows a wart on the hands and feet. Appearance of the wart depends on the HPV type and the infected site. Picture 2 is the condylomata acuminata. The lesion may appear as a soft, flesh-colored warts that are raised and cauliflower-shaped. Picture 3 shows the cervical cancer. Now, what is HPV? I believe you have got the answer. Let's go on to the next question. How is HPV transmitted? HPV is ubiquitous and resistant to inactivation. It can be transmitted from unclean surface of countertops or furniture, bathroom floors, and towels. The individual acquired the virus 1. By direct contact through a very tiny breaks in the skin or mucosa. 2. During sexual intercourse. 3. When a newborn is delivered through the mother's infected cervix and vagina. There are some risk factors for HPV infection, including 1. Weakened immune system. 2. A breach in the skin. 3. Multiple sexual partners. 4. Tobacco use. Now then, the third question. How can the infection be diagnosed and prevented? A few approaches are available. Cytologic changes can be detected. Histological appearance consisting of hyperplasia of the Pringle cells and a sex production of keratin are characteristic for warts. The presence of a koilocytotic cells in pap smear indicates HPV infection. DNA molecular probe PCR analysis of cervical swabs are usually chosen for the diagnosis and typing of the HPV infection. After the diagnosis, what shall we do next? In fact, there is no cure for HPV. Most are cleaned up by the immune system without any treatment. Treatment is used to relieve or manage the symptoms of the disease. For example, warts can be removed by a cryotherapy, and surgery may be necessary for the removal of laryngeal papillomas. How to prevent HPV infection? There are some effective measures including 1. Avoid direct contact with infected tissues. 2. Adequate precautions to prevent sexual transmission. 3. Routine pap smear for sexually active women. 4. HPV vaccination. Currently, the vaccines containing the L1 major capsid protein assembled into virus-like particles are used for immunization. A 4-valent, a 9-valent, or divalent HPV vaccine is recommended for girls and boys starting at age 11 years before sexual activity to prevent cervical cancer, penile, and anogenital warts. Although the vaccine is effective for prevention, it does not offer protection against all possible HPV strains. Finally, we go back to the question of the caves. Trust you can answer them correctly. Thank you for joining us. Next time, we will share some other interesting contents. Here are the references.